Method of localization and radio labeling. Things to look for in an ideal radiopharmaceutical: ease of availability, short effective half-life, particle emission, photon energy, high target to non-target activity ratio. Availability. It should be easily produced, inexpensive, and readily available. It needs to be available in a reasonable amount of time. Distance between the user and the supplier limits the availability of short-lived radiopharmaceuticals. Tech can have a generator on site and just get tech from it. Physical half-life. Time span of a radionuclide decaying, independent of physiochemical conditions, characteristic for a given radionuclide. Biological half-life. Time needed for half of the radiopharmaceutical to disappear from the biological system. Radiopharmaceuticals disappear from the system through such mechanisms as urinary excretion or perspiration. Effective half-life. Want a short effective half-life. Net or effective rate of the loss of radioactivity related to the physical and biological decay constants. Half-life calculation. 1 over the effective half-life is equal to 1 over the physical half-life plus 1 over the biological half-life. Particle emission. Radionuclides decaying by alpha particle emission should not be used as a radiopharmaceutical. Particle emissions cause more damage to the tissue than gamma rays. The decay method. The ideal method of decay is electron capture or isomeric transition. It does not emit particles, therefore it reduces the dose. The preferable energy is between 30 and 300 keV. High target to non-target ratio. It is desirable that the radiopharmaceutical be localized preferentially in the organ under study. You don't want non-target areas to obscure the structural details of the picture of the target organ. We want most of our radiopharmaceutical in the target, not the surrounding tissues. How much radiopharmaceutical that will go to the target depends on concentration, blood flow, and method of localization. The concentration of the tracer in the blood flow to the site, and the effectiveness of the method of localization. The method of localization, the definition is the route that a pharmaceutical takes. Another term is also biorouting. Most radiopharmaceuticals use a combination of more than just one type of localization. Capillary blockage. Intentionally blocking the capillaries with particles. We are creating microemboli. 3 important aspects of particles: the size, the hardness, in order to say, I was just talking about that. Composition. Example would be a lung perfusion with MAA. It temporarily blocks the capillaries in the lungs. Phagocytosis. Physical entrapment of colloidal particles by the Kupffer cells in the reticular endothelial system. An example would be the TEC sulfocolloid for the liver spleen scan. RES is part of the immune system. RES stands for reticular endothelial system. The Kupffer cells in the liver, spleen, and bone marrow function to trap and dispose particles. The particle size is important. The smaller the particles, the greater the bone marrow uptake. Larger particles tend to localize in the liver and the spleen. Cell sequestration. Injection of damaged red blood cells to produce images. An example would be technetium-99m RBC or chromium-51 for spleen imaging. The spleen's function is to dispose of damaged red blood cells. Therefore, we damage them using heat and then label them. Active transport. The use of the body's normal operative metabolic pathway in for moving a radiopharmaceutical across a cell membrane and into the cell. In this case, the drug has an affinity for the target organ or system. An example would be iodine-131 for thyroid imaging and uptake. Iodine is utilized by the thyroid to create hormones. If we utilize radioactive iodine, it will be taken up by the thyroid. Thallium-201 is uptake in the myocardium. Receptor binding. Binding of a radiopharmaceutical to high-affinity receptor sites. Receptor sites are proteins on the cell membrane or within the cytoplasm of the nucleus that bind to a specific molecule. An example would be neurotransmitters or hormones for tumor imaging. An example would be indium-111 octreotide for localization of neuroendocrine and other tumors based on binding of a somatostatin analog to receptor sites in tumors. Technetium-99m abscitide for DVT. Abscitide is a synthetic peptide that binds to glycoprotein found on activated platelets. These are activated when there is an injury to attract fibrogen to help form the clot. Abscitide binds to peptides that are on activated platelets and allows for imaging. Compartmental localization. The placement of a radiopharmaceutical in a fluid space and maintaining it there long enough to image that fluid space uses the body's boundaries and parameters. An example is xenon-133 lung ventilation studies, or technetium-99m red blood cells used in the gated blood pool studies, MUGA. Simple or passive diffusion. Diffuses across the cell membrane and attaches to the cell component. Active diffusion doesn't attach. This is passive diffusion. So passive diffusion diffuses across the cell membrane and attaches to a cell component. It is also called exchange diffusion. Examples would be technetium-99m sestimib for the cardiac perfusion imaging. Technetium-99m sestimib crosses the cell membrane of the myocyte and binds to the myocardial mitochondria. Chromium-51 for the red blood cell survival time study. The chromium will cross the cell membrane and bind to the hemoglobin. Technetium-99m DTPA in brain imaging. Again, the DTPA for aerosol imaging. Xenon-133 for the lung ventilation imaging, and indium-111 DTPA in cysternography. Chemotaxis. Process of leukocyte migration to the site of injury. An example would be indium-111 labeled white blood cells to localize infections. Antigen and antibody reaction. Monoclonal antibodies. We are labeling the antibody that will seek out the surface antigens on tumors. High specificity. Examples: iodine-131, indium-111, and technetium-99m labeled antibody to localize tumors. Indium-111 OncoScan, a monoclonal antibody specific tag 72.3, a tumor-associated glycoprotein expressed by colorectal and ovarian carcinomas, has been very useful in evaluating recurrence of these diseases. Chemisorption. Drug will chemically bond to the target, aka physiochemical adsorption or ion exchange. An example would be technetium-99m MDP used in bone imaging. It binds directly to the organic components of the bone. Uptake of technetium-99m phosphonate complexes in bone. Phosphorus will go to the bone that's repairing and get ion exchange. Metabolism. Radiopharmaceuticals that work with the body's metabolizing glucose. An example would be fluoride 18, FDG, uptake in the myocardial and brain tissues. Factors influencing the design of the radiopharmaceuticals: compatibility, stoichiometry, charge of molecule, size of molecule, protein binding, solubility, stability, biodistribution. Compatibility. When you are about to prepare a kit, the first thing you need to consider is whether the labeling process is possible. Only specific radionuclides label certain compounds depending on their chemical behavior. For example, indium-111 tagged with DTPA is formed and remains stable for a long period of time. Stoichiometry. You need to know the amount of each component to be added. Concentration of technetium-99m in the eluate is about 10 to the negative 9th exponent m. Chelating agents, DTPA or EDTA, used to ensure bond formation. A high or low concentration of any one component may sometimes affect the integrity of the preparation. Charge of the molecule. Charge determines the solubility in various solvents. The greater the charge, the higher the solubility in aqueous solutions. Nonpolar molecules tend to be more soluble in organic solvents or lipids. Polar molecules are soluble in water. The size of the molecule. Size is an important determinant in the body molecule's absorption in the body. Larger molecules are not filtered by the kidneys as easily. Protein binding. Regardless of whether a drug is radioactive, all drugs bind to proteins at various levels. Indium, gallium, and other metallic ions bind firmly to the transferrin in plasma. Protein binding affects tissue distribution and plasma clearance of a radiopharmaceutical and its uptake by the region of interest. Solubility. The radiopharmaceutical should be in an aqueous solution at pH which is compatible with the blood's pH. The blood's pH is 7.4. The cell membrane is primarily composed of phospholipids, therefore a radiopharmaceutical needs to be lipid soluble in order to diffuse through the cell membrane. Lipid solubility of a radiopharmaceutical is a major determining factor in the localization in an organ. Solubility. The higher the lipid solubility, the greater the diffusion through the cell membrane, and hence the greater its localization in the organ. Protein binding reduces the lipid solubility of a radiopharmaceutical. An example is indium-111 oxide is extremely soluble in lipids and therefore used for labeling leukocytes and platelets. The stability of a radiopharmaceutical is a major concern in labeling. It must be stable both in vitro and in vivo. In vivo, breakdown results in undesirable biodistribution such as increased background activity. Temperature, pH, and light are factors contributing to in vitro breakdown. Biodistribution. The study of radiopharmaceuticals' biodistribution is essential in establishing its usefulness. Biodistribution includes tissue distribution, plasma clearance, urinary excretion, and fecal excretion after radiopharmaceutical administration. Methods of radiolabeling. There are 5 major methods employed in the preparation of labeled compounds for clinical use: isotope exchange reactions, introduction of a foreign label, labeling with bifunctional chelating agent, biosynthesis, and excitation labeling. Isotope exchange reactions. In this type of reaction, one or more atoms in a molecule are replaced by isotopes of the same element having different mass numbers. These labeling reactions are reversible and useful for labeling iodine-containing material with iodine radioisotopes. Examples are iodine-125, labeled T3 or T4. Introduction of a foreign label. Radionuclide is incorporated into a molecule that has a known biological role. The tagging of a radionuclide is foreign to the molecule and does not label it by exchange of one of its isotopes. The chemical bond is formed by chelation of a transition metal. Chelation. More than one atom donates a pair of electrons to the foreign acceptor atom. An example: all technetium-99m radiopharmaceuticals, indium-111 and fluoride-18. Labeling with bifunctional chelating agents. This chelating agent is conjugated to a macromolecule on one side and to a metal ion by chelation on the other side. There are 2 methods, indirect and direct. The direct methods involve several steps, therefore not producing an ideal yield. Due to the presence of the chelating agent, the biological properties of the labeled protein may be altered. Examples are indium-111 DTPA or technetium-99m DTPA. Biosynthesis. A living organism is growing in a culture medium containing the radioactive tracer, which is later chemically separated. For example, vitamin B12 is labeled cobalt-57 by adding the tracer to a culture medium in which the organ Streptomyces is grown. Streptomyces. Recoil labeling. Limited interest since it is not used on a large scale for labeling. General idea. Particles emitted in a nuclear reaction are emitted from the nucleus and recoil atoms are produced which form a bond with other molecules. The downfalls. High energy of recoil atoms results in poor yield and lower specific activity of a labeled product. Excitation labeling. Utilizes radioactive daughter ions produced in nuclear decay process. Energetic charged ions are produced that are capable of labeling various compounds of interest. Examples: xenon-123 decays to iodine-123. The yield is very low with this method of labeling. Important factors in labeling: efficiency of labeling process, chemical stability of the product, denaturation or alteration, isotope effect, carrier-free, storage conditions, specific activity, radiolysis, purification and analysis, and shelf life. Efficiency of labeling process. Ideal to have a high labeling efficiency, but sometimes lower labeling efficiency is acceptable if the product is pure and not damaged by labeling method. Chemical stability of the product. Stability is related to the type of bond between radionuclide and the component. Stability constant that is a large for a labeled product means that there is a greater stability. Denaturation or alteration. Alteration of structure and or biological properties of a labeled compound due to physicochemical conditions during the labeling process. Example: red blood cells denatured by heating. Isotope effect. Results in different physical properties due to the differences in isotope weights. Not as serious as an effect with heavier isotopes. Carrier-free. Radiopharmaceuticals tend to be adsorbed on the inner walls of containers in a carrier-free state. Therefore, techniques have been developed in which labeling yield is not affected by the low concentration of the tracer in a carrier-free state. Storage conditions. Many labeled compounds are susceptible to decomposition at high temperatures. Albumin should be stored in the refrigerator. Light can also break down components, therefore kits should ideally be stored in the dark. Specific activity. Activity per gram of the label material. In some instances, high specific activity may be required in radio-labeled compounds, and other times, high specific activity can cause radiolysis and should be avoided. Radiolysis. Labeled compounds which are decomposed by radiations emitted by the radionuclides present in them. The higher the specific activity, the greater effect of radiolysis. Radiation may decompose the solvent, producing free radicals that can break down the chemical bond of the labeled compound. Purification and analysis. Radionuclide impurities are radioactive contaminants that arise from the method of production of radionuclides. Fission in a reactor is likely to produce more impurities than nuclear reactions in the cyclotron due to the fission of heavy nuclei. Radiochemical and chemical impurities arise from the incomplete labeling of the compounds, and the amount can be estimated by running quality control tests. Shelf life. Period of time in which the labeled compound can be used safely. The loss of effectiveness of labeled compound over a period of time may result from decomposition depending on the physical half-life. Usually a period of 1 to 3 physical half-lives or a max of 6 months is the recommended limit for the shelf life of a labeled compound. Technetium labeled compounds vary between 0.5 to 18 hours. Labeled with technetium-99m. Favorable physical and radiation characteristics. Readily available in sterile, pyrogen-free, and carrier-free states from the Molytech generators. Pyrogen is a substance inducing fever. Transition metal. Atomic number is 43. No stable isotope of technetium exists in nature. There are 8 oxidation states which exist. +7 and +4 states are the most stable. Freshly eluted tech has an oxidation state of +7. Tech needs to be reduced to a lower oxidation state in order to be able to be labeled. Reducing agents are commonly used. Stannous chloride, stannous citrate, stannous tartrate, concentrated hydrochloric, and ferrous sulfate. Tin in concentration hydrochloride is also added to ensure reduction. You inject pertechnetate, not free pertechnetate. Know the difference. Nitrogen gas or antioxidants are added to prevent oxidation of kits, which cause the presence of free pertechnetate. Hydrolysis of reduced tech. Possibility that reduced the tech may undergo hydrolysis in aqueous solution, which will cause interference with exam. Can add enough of chelating agent, MTP, DTPA, etc., to overcome hydrolysis. When preparing technetium-99m labeled compounds, 3 species may exist. Free tech that has not been reduced by tin, hydrolyzed tech that did not react with chelating agents, or bound tech chelate, which is the desired compound formed by the binding of reduced tech to the chelating agent. Oxidation states of tech in the free tech radiopharmaceuticals. Tech 7+ is pertechnetate. Tech 5+ is tech with EDTA, HMPAO, and gluciptate. Tech 4+ is DTPA, EDTA in alkaline and neutral solutions. Tech 3+ is DTPA, EDTA, DMSA, and HIDA in acid solutions. Tech 1+ is sestimib. Kits for technetium-99m labeling. Colloids and labeled particles. Stabilizing agents added to the colloidal preparations to prevent clumping. Examples include gelatin, polyvinyl pyrrolidone, or carboxylmethylcellulose. Example of colloid is technetium-99m sulfocolloid. The sizes range from 0.5 to 5 micrometers. An example of a larger colloid is TEC-MAA, which the sizes range from 15 to 100 micrometers. Kits for technetium-99m labeling. Additives and preservatives. Added to the radiopharmaceuticals to prevent degradation by radiolysis or bacterial growth and to maintain a proper pH. Preservative can function as a stabilizer and antioxidant or bacterial agent. Stabilizer added to maintain integrity of the radiopharmaceutical in its original state. Examples include ascorbic acid, gentisic acid, citrates, and acetates. Bacterial agents used to prevent bacterial growth. Examples include benzyl alcohol or ethanol. The end.